नमस्कार विद्यार्थी मित्रांनो. Today we shall learn the transport of respiratory gases and oxygen dissociation curve or Bohr effect. First of all, we should know exchange of gases which takes place in our body in two different places. First, already you have learned how that exchange of gases takes place in the lungs. It's a very simple mechanism. But whenever that exchange of gases takes place in the alveoli and blood, then that is called as external respiration, or it is also called as lung respiration. And exchange of O2 CO2 also takes place in the tissues. So that type of respiration is called as internal respiration or tissue respiration. So let us see how the exchange of gases takes place in our body. As you know, whenever we inhaled air, it has oxygen, and that partial pressure of oxygen is about 159 millimeter of mercury. Whenever I am talking about the partial pressure, you must know partial pressure means the gas created pressure in a mixture of other gases also. So here, partial pressure of oxygen in alveoli, it is about 159 mm of Hg. And what about the partial pressure in venous blood, which comes in the capillaries of alveoli? It is about 30 to 40 millimeter of mercury. So there is a tremendous difference in pressure. So what happened here? Due to differential gradient, simply diffusion takes place, and by the process of diffusion, oxygen combines with the hemoglobin. As we know, hemoglobin is having much affinity with that of oxygen, and thus there is a reaction, and by that way here hemoglobin becomes oxyhemoglobin. As we know, hemoglobin molecule is found in the form of Hb4, so it requires 4 oxygen molecules, and thus there is a formation of Hb4O8. This is the saturated oxyhemoglobin. Exactly reverse reaction takes place in the tissues, and hence this reaction is reversible. When oxygenated RBCs, when that oxyhemoglobin reaches to the tissues, here it liberates oxygen wherever necessary, and Hb remains free. It combines with H plus and CO2 also. It come back to the heart. That rest of the details we shall learn in the next lecture. But here, whenever oxyhemoglobin reaches to the cells, it is responsible for releasing the oxygen. In the same manner, what happens on the surface of alveolar air, the partial pressure of CO2, it is about 46 mm of Hg in case of venous blood which come into the capillaries. And in alveolar surface, partial pressure of O2 is about 40 mm of Hg. Here also, due to the differential gradient, here CO2 liberates, here it has more pressure, more partial pressure. So it diffuses into the alveolar air, and by that way in expiratory air CO2 is discharged out. So by this way exchange of gases takes place on the surface of alveoli due to the pressure, partial pressure difference. Now again, one important point you have to note: hemoglobin has ability to take that oxygen, but hemoglobin always bind with that of oxygen very loosely, so that whenever it is necessary to the cells, it is get liberated. So by this way, about 99% of the oxygen is carried by the hemoglobin, and a very less amount, means about 0.3% only, or only 0.3 ml of oxygen per 100 ml of blood is carried by the plasma, and 20 ml of O2 is carried by hemoglobin per 100 ml of blood. So let us see how that oxygen is carried by hemoglobin, and thereafter how it is get dissociated into the required field, and it is explained, very well explained by different scientists. But here Bohr explained by a very different way. म्हणून आपण बोर इफेक्ट शिकतो. एक डॅनिश फिजिओलॉजिस्ट होता. या डॅनिश फिजिओलॉजिस्टने 1904 मध्ये ही थेअरी मांडली, आणि त्याच्या म्हणण्यानुसार जेव्हा जेव्हा CO2 आणि H+ आयन कॉन्सन्ट्रेशन वाढलेले असतील तेव्हा डिसोसिएशन वाढतं. ही त्याची थेअरी आहे, हे त्याचं प्रिन्सिपल आहे. मग आता आपल्याला बघायचं आहे की या ठिकाणी डिसोसिएशन कर्व काय भानगड आहे. अगोदर तुम्हाला हा ग्राफ समजला पाहिजे. इट्स अ व्हेरी सिम्पल मेथड. अगदी सोपा असा हा ग्राफ आहे. या ग्राफमध्ये काय दर्शवलं आहे बघा. एका बाजूला percent saturation with oxygen with that of hemoglobin and partial pressure. एका ॲक्सिस वरती percent saturation of oxygen with that of hemoglobin against partial pressure of oxygen which is plotted here. आणि यामुळे बघा तुम्हाला अगदी सहजगत्या लक्षात येईल, जसा जसा हा प्रेशर वाढतो तसं हे सॅच्युरेशन वाढत जातं, आणि जवळपास 100 mmHg ला this graph reaches to 100% saturation. It shows 100% saturation like this. So it forms such kind of sigmoid shape. It forms sigmoid shape like this. But according to Bohr, here whenever H+ ion concentration increases, that graph is shifting to the right side, or when CO2 increases, dissociation also increases. It's a very simple mechanism, and we know whenever we are doing any work, suppose that I am doing work with this hand, here there is a need of energy. Ultimately, by utilizing that energy resources, here CO2 will be formed. That CO2 is the byproduct of that reactions, and that CO2 it dissolved in the solution which is present around the cells or into the protoplasm also. So here when that CO2 is get dissolved with H2O, then it forms carbonic acid H2CO3. Now that H2CO3 is responsible for releasing H+ ion concentration into the solution. So that definitely pH has to be changed. If pH is changing here, definitely dissociation has been disturbed. How is that? Let us see. Suppose that CO2 is more, so H+ ion concentration it will be again increases, so that pH changes, and that if pH changes, then it will become like this. Here this graph is shifted to right side when pH is 7.2, means here pH decreases. When H+ ion increases, pH decreases, right? Now what about the H+ ion concentration? If it decreases, then what happen here? Here that graph try to become T-shaped one like this. Means when pH increases, if suppose that it reaches to 7.6, then you will get this kind of curve. So here this oxygen dissociation curve is always shifting as the pH changes. So you can say when pH decreases, this graph is shifting on right hand side, and if pH increases, here pH increased from pH 7.4 to 7.6, what happened here? This graph is shifting to left side. What about the CO2? When CO2 increased, it has to be shift on the right hand side. When CO2 decreased, it shifted to left side. Now again, some other factors are also responsible for dissociation curve. Which are they? Temperature. Temperature is also a very important factor which is responsible for fluctuating this graph. Actually, it is not too much, but it is still noticeable in case of hyperthermia and in case of hypothermia. Then different metabolites are also there in our blood. Which metabolites are there? 2,3-diphosphoglycerate is present in our blood. If it increases, graph is shifting to right hand side. If it decreases, DPG, then it shifts to the left side. In the same manner, electrolytes are also responsible for shifting this graph. So these are different factors which are responsible for the dissociation of oxygen. This effect is called as Bohr effect, or it is also called as oxygen dissociation curve, or it is also called as oxygen hemoglobin dissociation curve, or it is also called as oxyhemoglobin dissociation curve, or it is also called as Bohr effect. I hope that you might be understood. If you have any doubt, you may ask me at Dr. Kiran Ahira at gmail.com.